We had seen then that the proteins are all of the alpha type of the series L and of our stereochemistry S, except glycine and cysteine, and we are going to draw those two amino acids to understand why it says here, except glycine and cysteine. We have that the following is the structure of glycine. And the following is the structure of the cysteine. And it said cysteine, right? Yes. I also had a doubt with the methionine. This is cysteine, and this is glycine. We said that all amino acids are of absolute configuration S except these two, because what can you tell me about glycine, the first? No, good observation, is very subtle. The difference to the Nina is H3, that would be to the Nina do not, right? That would be the Nina with a methyl in that position. But what we have here is, and they are suitor ions, okay, if they are all suitor ions. Because the net load is 0, but there are internal loads, well, because this is not S. There is no central chiral, very well. Then we are talking that this is chiral. It is the only amino acid that is chiral that does not prevent it from being able to adopt a space arrangement of type appropriate for the interconnection of the amino acids. And this other is nothing more than for a question of zip rules. Remember that to assign chirality, we apply the zip rules, the rules of rules. Can Ingo prelude. And if we apply those rules that are by atomic weight, the first priority. Here is for nitrogen, the second priority is for, and here there is a carbon-carbon tie, but then we have to see what follows. That would be sulfur, that has greater atomic weight than oxygen. Therefore, apparently, it is S. But since the proton is horizontal in the feature projection, in reality, the center of absolute configuration R. But that does not imply that it ceases to be of type of the series. That series was this of feature when the amino is on the left in the most oxidized projection, of the most reduced below the relative configuration. Also we have to explain that, because glycine and cysteine, one is chiral and the other is of type R. I have a doubt with the methionine for this. We are going to go back to this image. I developed amino acids because the methionine, to see where the methionine is not, the methionine has one more carbon. If you look at it, therefore, in priority, it will win up here, because first it has oxygen, and then here there is carbon, so we have that it is with a carbon found with an oxygen, so this is still of absolute configuration. Well then, we return now to the appointment to continue delivering information about amino acids. The amino acids that constitute proteins we already saw in the last. All of type alpha, the alpha carbon, is the one that contains the amino acid residues. The residue that makes each amino acid different, and based on the residue, is that we are going to classify the amino acids in different families, and those families have to know them. They have to understand which is the basis of the classification, because that will allow them to understand intermolecular interactions and ask how many amino acids the proteins make up. 20 very well, and we are going to classify those 20. And we had already explained the previous class, or if we had not said it, we mentioned it now. That amino acids are two functional. They present a basic group, but that is going to be if it is isolated. By the general it is protonated, and they present an acid group that if the amino acid is isolated. By the general, I am going to explain why I say by the general it is ionic. The carbon-carbonylic is the electrophilic center, while the nitrogen is the nucleophilic center. And you will say, but how is it going to be a positive nucleophilic nitrogen? Because the protein synthesis is not carried out mid-acquaintance, when this is only so ionized, but within the cytoplasm we are in mid-acquaintance, but the organelle, where the protein synthesis occurs, is ribosomes. There the concatenation occurs with enzymatic support. That is, it is mediated by a molecular machine. The nucleophile is the amino group when it is neutral for fair. Ribosome concatenates it, and the electrophilic is the carbon-carbonylic, and this by amino acids to concatenate with themselves well. We are understanding the amino acids. Which of these that are down here is the amino acid? Or they are amino acids, those who have an amino group or only one of them. Exactly, this is amino acid. Well, then we have that this is the alpha carbon, and we have the carboxylate, the ammonium group. And we have that this differentiates it. It is a methyl group, therefore this is alanine. Okay, it is the alanine now. How do we know that it is L? Because in a picture projection, if we draw the most reduced down, it will meet what we just explained about the amino group. Will remain to the left. Okay. Do not forget that the amino acids are of type L configuration. Here we see that this is priority 1, 2, and 3. The of absolute configuration. Yes, okay, but there is no interrelation between it be L and that the chiral center. Is that and the levohero or dextrose? We also saw the last class that other concept levohero. Or dextrose that had to do with polarized light. There was no way to predict it. Remember that there are three independent things: absolute configuration, relative configuration, and the optical property or optical activity of the compound. There is no interrelation between them. Predict from one property to the other. It is probably dextrose. I do not know. I look for it, but there is no way to predict it well. The one that follows here is not an amino acid, but that these three molecules are characterized because all of them, we are going here, these four molecules have a similar molecular weight. So when the molecules have similar molecular weights, one expects that their physical chemical properties in some aspects are similar. But we see that, for example, the alanine, which is the amino acid, and that like a suitor ion has very different properties. For example, the alanine presents a solubility in water that is not complete. Then it does not present complete solubility, contrary to what common sense can tell us, because one tends to think that the suitor ions of water, not necessarily in this case, we see that its solubility is not complete. What affects drastically the properties of the amino acids is the group R, the amino acid residue. What other particularity do we see of the alanine that behaves like a salt in terms of its salt melting point? More than 300 degrees Celsius has a very high melting point to make organic molecules. Organic molecules have relatively low melting points for this side. This has a strikingly high melting point, given that it is an internal salt, a suitor ion. And remember that the salt is like sodium chloride. In general, they have high melting point. What is the melting point of sodium chloride? Someone has any idea? Very high. Well, if we compare, for example, the melting point of glucose, glucose would be 6H12. Or 6, a large but organic molecule, its melting point must be less than 100 degrees Celsius. How do we know that? Or close to 100? Because we can make candy. In the pan, it melts easily. But if you put salt in a pan, it will not melt. Its melting point is around 800 degrees Celsius. Then we see that the amino acids have a behavior like kind of salt in terms of melting point, what modulates the behavior. And that is what I want you to say from here, because we are seeing this, because what modulates the behavior of an amino acid for practical effects is the residue. Okay, don't forget it. And that's where we have the 20 amino acids that constitute the proteins of the human body. So, how are we going to classify this? We are going to classify it according to the residue. Residue is only a proton here. The residue is a methyl then that already speaks to tight residues. They are very good. And that is a good observation, not ionizable. The residue is not ionizable, and on the other hand, the residue is hydrophilic or hydrophobic, well hydrophobic. Here we have the abbreviation of 3 letters and the one. Let's go now. There are several like this, but I want to take a leap to others more now. Little further forward. And for example, if we maximize here, note the following now. We have the serine, the trine and the triosine are characterized because the residue this time is already H2 or H here. We also have an ethyl chain or H, but the point is that there is a functional group that gives it polarity. That is, these are again, they are neutral. To 7, and they are polar. When I say polar, they are hydrophilic. And then we already have 2 extremes to form proteins: lipophilic amino acids, hydrophilic amino acids, and the union of a new drug to the protein has to have both things, hydrophobic interactions and hydrophilic interactions with these residues that are going to be hydrogen bridges among other interactions. Now that is not all, because we have 2 more groups. What happens, for example, here with lysine and arginine? Perfect. There is an extra basic group in the structure. Then it would be this group here, and in the case of arginine, it would be the guanidine group. Then these would be with an extra basic group of basic amino acids. To see Pika Sua, Yusuf Piga. If it is basic, this is about 12,9, around 9. The value of pK sub A is around 9, and the arginine is still more basic, around 12. It is approximate in any case, and obviously we are not going to give exact values for each functional group. And there is another basic that I want to stop here a sec. Perfect. The histidine is also super important. This group for something that I am going to explain. Below it, it is also one of the basics. How is this heterocyclic called pyridine, the name. And imidazole. Imidazole. Then we have that in the case of imidazole, pK sub A, a value. Look at what happens, because I annoy you with this, because if we are at pH 7, this will be 100% protonated to pH 7, and the guanidine with greater reason. 100% protonated already is aromatic, is less basic, is around 7. That means that the imidazole can be 50% protonated, has a degree of ionization of 50%. Now, because that is important, because that implies that the imidazole can both capture protons, has a dual nature as to give protons. And that is tremendously important in what type of biological processes. When more specifically, when we have that the drugs are going to act in all receptors or in enzymes, and most of the time there are also other targets in enzymes at the level of enzymatic catalysis. This dual nature makes the imidazole be present in innumerable cavities of enzymes, because it can capture protons and then a proton transfer agent in enzymatic catalysis, and this is tremendously relevant in enzymatic catalysis. In the catalytic cavity of many enzymes, there are also receptors, of course. Then you have to be careful with the imidazole, is that the environment will determine if you prefer to be protonated or if you prefer to be neutral. Well, there we have already mentioned the 4 grades. I have a group left that would be these 2 here only. And here exactly we now have a residual chain with an extra acid group, aspartic acid and glutamic acid. Then these are the acids par excellence. And now the question is, and which group is more acid? The acid of the chain or the acid of the amino-acidic portion, properly such? That is the alpha amino acid, which is more acid, the alpha amino acid, or the acid of the chain. And the chain would be this one here. The alpha amino acid is that it is connected to the amino. Let's see, the acidity depends on the inductive effects. The amino group acts as an inductive attractor, making this link stronger or weaken if it is removing electron and is pulling the nitrogen electron, so it weakens and becomes more acid. Then usually the amino acid is more acid, or we are going to write it like this: it has a greater acidity. Well, you will have me. I have to learn all that more than knowing by memory. This is learning too. Reason. And for that is that this table here, we have the complete classification, been talking about and trying to understand rationally the 4 groups of amino acids that you have to know, because they are very important for a docking, for an interaction linking receptor to know the environment of the cavity, helps to design or better match a molecule to said environment. Okay, Then first, it says hydrophobic amino acids with a hydro nonpolar. We already have glycine, alanine, valine, leucine, isoleucine, proline. How are these two called cold? Very well, the eighth is pH phenylalanine, very well, but Christopher, you are trying to make a hydrogen bridge here for a while. That is very good. Perfect the famous tryptophan. Be very careful that this proton is not basic. Okay, ask, why is that amino group not basic? The electronic pair is 100% dislocated by resonance. Therefore, it is not basic, protonated, and it is classified as hydrophobic because the amount of carbon that this super high fragment does not achieve the NH increase the solubility. On the contrary, this fragment is hydrophobic I here. In the proline, which is an amino acid whose chain is cycled over the amino group. Of course, that is the proline if P is proline. Well, let's go then. There we have group 2 now. Look here, there is a small error in this table. And this one has to move it up. The methionine is bad here, and the methionine has to be up, okay, because the methionine has a chain that is quite lipophilic. This is an ethyl uncle, a fairly lipophilic functional group. So small error, this one goes up to the hydrophobic nonpolar. This is a fairly quite lipophilic chain. Even if it were oxygen, this would be super lipophilic. Well, and these then it says here they are non-ionizable to pH 7 because the amino groups are not protonated, because they are functions. What is the name of this? Amides and this other, because it is not ionized. And what sin does that have? And the alcohol is 16. Probably the ash is more or less than 16. Of course it is less than 16, because the anion stabilizes better on a sulfur and an oxygen, and there. Phenol stabilizes the charge by resonance. Then, what do you think the value? Less than 16 less is about 10, so it will be 100% neutral. I already point out this so that you remember that this will be 100% neutral to pH 7. It will not lose the proton. In some docking calculations that the programs sometimes leave us phenols with negative charge. Though this is wrong. Be careful, there should be a very particular amino acid that, or for example, I do not know to this, they also take out the proton and leave it as anion. It cannot, so be careful with. Well, and finally, here we have these two that I always confuse with the names D and the E of the shorter chain and the longer chain. Let's see it in the previous one, because thanks to the exact. And the other is the glutamate. As it is. If it costs similar question. Well, this was done, this of a letter was invented. In particular, because sequencing protein and writing with 3 letters was more cumbersome than in computer video, all the codes are written only in a letter, of course, as it is a question to see something I was going to ask here, these two. The aspartate and the glutamate, apart from being constituent amino acids of the proteins, at the neuronal level, They function as very well. They are also neurotransmitters. Excitatory and inhibitory, very well. In fact, in high concentrations of glutamate, it can become neurotoxic. Well, and finally, down here are the basic ionizable physiological pH. And I want to see how short-term memories are okay again. Jusip ki kaka. 8 no is very good. Exact I themselves molecule. And it 9 again. And this one here was more basic and very well 12. And the imidazole well. And those values. It is good to retain them. They are quite useful to understand the acid properties of molecules, the reactions that they are going to experience, etc. In fact, for example, at the island, when cataract processes occur, it is because certain lysines that are part of the human crystalline react this amino that loses the proton in a balance, reacts with what thing is elevated in a diabetes at the plasma level glucose. And this reacts with glucose form chip bases that are called, and in that way, the crystalline structure, crystallina producing cataracts, very well. Here I have this image there, and you all. The world knows this picture is not true, but it is altered not true, and because it is there it was, to illustrate some particular properties of other amino acids, such as cysteine there is. It was considerably more acid, 8.3, but pH 7 will be 100% neutral. Okay, well, when it loses the proton. We could not calculate what the ionization degree will be. I do not know if it is here. Next to it is not the ionization degree, 833 to pH 7. Okay then, as 90% neutral, yes, more or less. Yes, it should not be true. How much was the formula? Remember, the formula was what we place above. Because, in this case, it is behaving as acid above. Would be the form, the neutral form, or ionic of the chain, that is, the S is not true. I'm abbreviating it like this. And below the S, and that is equal to 10. If that careful, because it was not which was first, P at pH minus pK2. And see quickly, I am going to apply a calculator to see how much it gives us at this part. .0 approximately then 15 is 100%, and as we want the ionization degree that he called 6% would be that we place a 0.5, then the ionization degree. I'm going to erase here. It gave me 4 points, almost 5. Yes, 5% already. Around 5% is the ionization degree. The same is low. If it is being low, And so as you can see the difference, as it is a logarithmic scale, small changes close to 7 will generate abrupt variations. In this case, we are already moving away by more than 1 unit, so the ionization degree be mainly as she payoff physiological good, the cysteine that is here whose code of a letter is C. At the level of proteins generates folds of the secondary structure of a protein. I am going to explain that because between cysteines it can be formed exactly what is known as a disulfur bridge. Two cysteines can be interconnected through a process here to the right that happened. An oxidation occurred very well then to the right. We have an oxidation. Through oxidation, they are interconnected, altering the structure of proteins. That is, we can have two chains that approach the space, and when that covalent bond is produced, the shape of the protein changes because it has to be interconnected. Example of the disulfur. Well, obviously, the curly hair was here to illustrate this example. When someone has curly hair, it is because there is a large amount of disulfur bridge in the capillary protein, and when through a hair smoothing, one stretches the hair with an iron or whatever. This is broken through a reductive process that can be helped by heat, but another example would be this look. This that I am drawing here corresponds to the chain of an insulin. What is insulin? A neurotransmitter or a hormone perfect, and it turns out that it is synthesized from a gene. Before it, it was thought that they were two genes, so what I am going to explain now, exactly as it is the hormone, because it is the entire chain of insulin is synthesized by a gene. But here, between these two, or these two terminal parts that were close together, two disulfur bridges are formed. And in fact, there is more, but here, this would be the part that and then an enzyme comes. An enzyme that cuts this chain here and cuts here. It takes out a piece, and when taking out that piece, the final shape that one observes is like the insulin is formed by two chains that apparently came from two different genes. One sees two chains, one makes a gene. Another gene, no, but it was altered in a post-translational way. Then there was a post-translational enzymatic or post-transcriptional. What is the difference? Perfect, super, very well. And then we have down here to conclude. Ah, well, it was the case. Or phenol, also then for the least reason, because the phenol is going to be ionized and its pKa is 10.1, okay. It will be almost 100% in its neutral pH 7 form. Then it is very difficult to lose the proton in a biological cellular medium. Well, let's go down here. Let's go down, it says. Post-translational amino acids. Modified these amino acids that I am showing here being found in proteins, but they do not come from the diet. They do not come from food. For example, or phosphoserine, oxygen, or hydroxyproline. I need to define here the chiral centers, because the stereochemistry of the human body is not racemic. It always generates enantiomer. Then this is perfectly defined, only that here the stereochemistry that corresponds in each case is not put. And finally, carboxyglutamic acid that hasn't. None of these come from the diet, but once the protein was synthesized. Post-translational. When the process of protein synthesis ended, some enzymes modify certain amino acids in these specific positions so that the function of the protein carried out okay. So if there are other amino acids detected, but they come from a later modification. If they had to classify these, for example, this one here already with this term, this in which group? The first in which group they would classify it very well, acid amino acid, because this comes from a phosphoric organic acid type OK, and it will be 100% ionized to pH 7. It is so acid that it is always anionic. Finally, to conclude this introductory part of the amino acids, it says here important amino acid that are not found in proteins. Several are mentioned there. In particular, we could highlight some more, such as if we go here, beta-alanine, it says that it is part of some non-human natural peptides. D-alanine, good D-alanine is also neurotransmitter in the brain. They are, as neurotransmitters, per se, aminobutyric acid. If you remember this, last class, it is in the brain. It is a neuro. Of what type? It was inhibitory. Very well, this is the GABA. Glutamic acid, now you will say, but here it is of be careful with this look. If it is of glutamic, it can process human metabolism to form proteins. No, it is not part of human proteins. But yes, it is in bacteria. Okay. In fact, in the bacteria, we have that the glycan peptide of the bacteria there, bacterial glycan peptide, has a terminal portion of the type of alanine, and that terminal portion of the glycan peptide is imitated by the beta-lactamic agents. Look, I'm going to show you here because that's a nice example of application of. This should be here. I have all this scratched, but the idea is going to be understood. Look at the terminal portion of the pier. Glycan peptide of alanine is this one from here. When the bacteria is synthesizing its glycan peptide, there's a terminal portion of the type of alanine. Here are 3 dimensions. This here is an alpha carbon that has the amino that continues to the peptide by the peptidic bond. Carbon alpha has the carbon dioxide of the acid that is forming here. I'm going to explain more about this with another D-alanine, which is the one that continues here in a terminal way. Here is the alpha carbon. Here is the methyl of the alanine and the free carboxylate. Now, if you compare this portion with the beta-lactamic system of the antibiotic agents, such as, you can name me a beta-lactamic like any penicillin very well, as the mucicillin penicillin. In general, notice that the square ring that we had seen last class with the blue aren't well. I say that it is here. Next to notice how this part is identical. Carbon dioxide, nitrogen, carbon dioxide, and then certain is highlighted, but it is blue. Then comes carbon, and finally an acid. Then it imitates very well the portion of alanine, joining transpeptidases that recognize that region of the glycan peptide to concatenate it, and therefore the enzyme remains inhibited when joining the beta-lactamic system. And there is the importance then recognizing the amino acids. And if now we return to the point, let's go over to think that this would be open there. Yes. We have others more okay that are part of the tissue of animals, etc. but this interests me. L-tyroxine. Thyroid gland is the thyroid hormone. Then per se it is a hormone from which amino acid. This was synthesized very well. Then this comes from the tyrosine metabolism. And we generate the L-tyroxine because the configuration of Fischer, the amino group, is on the left, and notice that this compound is polyiodized. As far as I know, you correct me, it seems to me that in the human metabolism there are no proteins. With halogens such as chlorine, bromine or fluorine, I think there is not, but here we have with iodine but the hormone apart from the amino acid that circulates freely through the plasma. But its protein structure, there are no proteins that contain chlorine, bromine or fluorine or iodine. Modified post-translational, I think there is not. This would be like the example, okay, why I mention this, because it is good that the drugs have halogens so that those halogens can interact with the proteins very well, and what we are going to determine now that it would be the last regarding amino acids is what is known as the isoelectric point of an amino acid, the ionization state of an amino acid. Mathematically, we have that amino acids can be in forms not true. For example, in acid solution, it would be in a cationic form 100% protonated. When we have an amino acid in a very acidic medium, it will be cationic. In a basic alkaline medium, the hydroxide can capture a proton, and therefore it will be anionic. The question is, what is the intermediate point? In other words, to what pH the peptide amino acid or protein presents net load equal to 0. The answer is isoelectric point. The isoelectric point is the pH to which the amino acid peptide or protein presents a net load equal to 0. That is the isoelectric point, is a pH value. We're going to explain this, for example, for the amino acid alanine. Well, we have that if the pH is very high, that is alkaline, the alanine will be with a negative net load, but if it is very acidic in the stomach, it will be with a positive net. What pH is with net load? 0. That is, what pH is going to be this positive and this negative? Is that calculated to do that? We have to do the average of the two values of the amino acid mathematically. Question to see here. It gives me 6 for the case of alanine. That means that pH 6, the amino acid to pH 6, the amino acid is found. To pH 6, here it is negative, and here it is positive, so it would be to pH 6. That would be the value of the isoelectric point. And ask this value of 2.34, for which functional groups 2.34 is the value of pK sub. A very well of the carboxylic acid, and the other, close to 10, is the halo. Value of pK sub A of the amino group. Then the average of the two values simply made, and we have the isoelectric point. Complicate the analysis. I do not want to see curves or anything like that of more complicated issues, but this one has 3 functional groups. What amino acid? To see what is the alpha carbon of the structure 1, 2, 3, 4, there is a number. The 3 very well, and then their first is to understand the amino acid, that is the alpha. We therefore have that there is connected the acid group of the amino acid, and the amino acid there must be the name, and all the rest of the chain would be the residue, the amino acid residues. Then, with this methyl acid residue, because that is the residue, that amino acid was that. And what was the shortest chain of the aspartic, of the glutamic, of the aspartic? And let's see if thanks very well, excellent. Then this would be the aspartic. Therefore, this structure, as it is drawn here, would be the structure of the amino acid. When we are in an acid pH, for example, to stomach pH, the amino acid would be protonated. In the amino, protonated the alpha carboxylic, and protonated the carboxylic. Question is what pH exists as a suitor ion, as it would be a suitor ion as a starting point. The net load has to be. Therefore, they can be ionized, the three at the same time. No, we have to find the pH value that allows only two of them to be ionized, so that the net load is 0. And we have there the values. Schoff pK to adhere your reasoning is the following. Before you see the answer, they tell you that you have to average, so no more look. The reasoning would be first to identify the most acid group, then the first question that you make, which is more acid. Already perfect, already very well. This is the most acid because it has the lowest pH value. Therefore, conclusion, if it is the most acid, it releases the proton. And now you proceed to draw the structure, and they say the most acid releases the proton. Then I erase here, and if it releases the proton, what load will it have? Negative, -1. And the second question is, what is the most basic? Which is the most basic? The only basic that there is, very well, because in this case, the amino group is not the only base present in the structure, then this is going to be the one that captures the proton. Do anything because it is already drawn in its positive form. Okay, then we have that. That structure, as it has been drawn, would be the structure of the suitor ion to point, and its electric point in the country. What is the utility of this? That one can apply this concept of electric point to peptides and proteins. And what is that? For you know, so that what is the grace of, for example, to know the electric point of a protein, Christopher, of course it serves to separate proteins. And we go there because this is not only a theoretical class, but there are also applications like this. It serves to separate peptides or proteins through electrophoresis in gel. Then we have here a gel electrophoresis. And this is not only applied to electrophoresis, but to any other principle of proteins, where they migrate by differences of polarities and by electrical differences. By capillarity, it can also be clear. In fact, in a Western blog, it goes up almost somehow. In a plate, it goes up, and it has a negative charge, as it will be the charges of the proteins, and perfect. But if they are all negatively charged, and an electric field is applied, the same is applied, then the principle is sufficient. And look here in the center, one shows the problem sample. We have 4 amino acids that we want to separate. We show them obviously. This is drawn illustratively, like there are 4 spaces to be able to separate, and the different isoelectric points of the amino acids of the problem mixture. Then once they are sewn, an electric field is applied. We therefore have here a cathode or negative electrode, and on the other side an anode or positive electrode. And the gel where they move has a fixed pH then. This gel has a pH of 6. That means that it coincides with the isoelectric point of two amino acids. If we go to the structures that are up there coincides with the electric point, this pH of 6 coincides with the isoelectric point of isoleucine and coincides with the isoelectric point of alanine. Therefore, to this pH isoleucine and alanine, that net charge, they have 0. And if they have 0 charge, they do not migrate neither positive nor negative. And they are in the center, but to pH 6. And there we go up to the structure of arginine. To P6 arginine will be protonated. What net charge it has? Positive. It will migrate to the cathode. While the aspartic acid to pH 6 has a negative net charge, and therefore it will migrate to the anode. Then that is the underlying principle to separate, in this case, by differences of isoelectric point. Well, having said this now, we can conclude this part with the various structures that proteins have, and for this we are going to define the connectivity that is given between, called the peptidic link. We are going to maximize this part here, because the proteins are formed by peptidic links, because they are not formed, for example, by a link of simply a link of type ether, a link of type ester that is also similar and easier even to form, because it is not formed by another link or interaction. Well, one of the reasons is that peptidic links here, there is one, are extremely strong. This functional group that you see here is an amide, functionally speaking. Then I am going to mark the peptidic link, that is the formation of an amide between the carboxylic acid of an amino acid group and the amino group of another amino acid question in this structure. That is being shown here, how many amino acids they manage to detect, that they are interconnected inside each other in this chain very well, because there are 4 alpha carbons. The alpha carbons are not true, they contain the residues. So this one here is an alpha carbon. It has an amino group, and it had an acid, but it was chained with the amino group of the other amino acid. Then, it is not true this group was also united here. There is another peptidic link with the amino of another alpha carbon, and the same here at the end. How many peptidic links count to? 4. There are 4 amino acids, but 3 peptidic links, then what we mark in C. Light blue are the peptidic links. Notice that here at the end there is no peptidic link. If the LOX were all neutral, this is the Suter ionic neutral form. Well, properties of the peptidic link. Well, the properties of the peptidic link are that, first, it is extremely strong because it presents a perfect resonance of the amino group towards the carbon, and that is illustrated below then. I want to show it here because that resonance is going to translate into something that I mentioned to you at some point exactly very well. Then here is the resonance we have, that this generates that the peptidic link has a double link character. Then stronger second observation to see Anton and the alkenes are rotable. Not true then. A plane is generated that contains 6 atoms, the oxygen, the carbon carbonylic, the nitrogen. proton and the alpha carbons of one amino acid and another amino acid. Remember that those are there. First, there is an alpha carbon that is part of those amino acids on those sides that implies peptidically speaking, two things. First, the disposition of oxygen with the proton is always transoid. And second, that we have a total of 6 coplanar atoms. Question: can there be a protein that the proton leaves for the other side? That the proton is looking towards here and the R down? Exactly, it is not adopted. Always the transoid disposition with the proton down and the oxygen up. On the opposite sides, probably for a hysterical reason, because the groups are larger and they are going to get in the way. Well then, that is an observation of the peptidic link that extremely strong, is of transoid type, and generates 6 coplanar atoms, and that union, remember that union occurs in the ribosome, who was the electrophile, the oxygen, the carbon carbonylica. And then, this is the electrophile center, and the nucleophile, the nitrogen atom. Then, the nitrogen atom nucleophile, the carbon carbonyl, electrophile, and this natural union only occurs mediated by enzyme ribosomal enzyme. That is, it is impossible for a protein to synthesize itself spontaneously in the cytoplasm of a cell. And there we enter, disturb the minds of many people through the scientific time, because it escapes from a naturalistic framework of interpretation, and before that, because a protein to be synthesized requires the pre-existence of another protein of the ribosome, then the question is how the first protein appears. So to speak, the proteins are not synthesized. There is no method or mechanism from which they can be obtained spontaneously in the cytoplasm of the cell or in the environment. They only proceed from the ribosomal machinery present in a cell. That is, the proteins require the pre-existence of proteins to exist. It is really surprising, and that can lead us to a discussion or conversation that already escapes from poorly naturalistic analysis of what the molecules are. Very well, and now if we look at the credits plans, see here they are, and we have this, that the peptidic link is not rotable. The peptidic link is not rotable, it is fixed, there is no rotation. Yes, because if not, how would it be if this was always rigid? All the flat? They would be flat chains. Then one says, how do you make such varied shapes? Well, it is because here, where the alpha carbon is of an amino acid, it is not true that it has its amino group and its carbonyl group. This alpha carbon does have the rotable link to the right and the rotable link to the left. Therefore, the planes that are like this can make this movement, and throughout the chain, that movement gives rise to the form, now the form of the protein that depends on the sequence of amino acids that you have. If you alter the sequence, it is throughout space, so the form, the information for that form that you have to adopt is encoded in the sequence of amino acids. That is, it is implicitly in the information contained in the chain, and those rotation angles that I am showing here have a name because this rotation for one side and this other rotation for the other side have a name. Let's go over there to look analysis of this before moving forward, and this is the next one, and here it is. Let's see it here. First there is the alpha carbon, the rotable link to the nitrogen is called very well. Then this is the letter fin. And for the other side between carbon and carbon, it is called no. Either it is, and here then we have the rotable angles phi and yes, and that rotation then the one that gives rise to the three-dimensional structure of the proteins, which are therefore the levels of the organization of the proteins. And what they depend on, well, they depend on the, and we are going to say this first before moving forward. Allow me to apologize and return me, but it is for you to understand this first observation in the peptidic link we have. That, for example, here where the peptidic link is here, there is a peptidic link, as you see in the peptidic link, that we had said that nature is like an alkene. So if it is like an alkene, the angle is close to an alkene 122 degrees, an alkene 120, then the link angle is close to the link angle that has an alkene 122 degrees, instead of the alpha carbon that geometry has. That molecular geometry has this carbon. This is not trigonal. There is a proton there that is not. Drawn it is a tetrahedron, and the tetrahedron 109.5 is the perfect angle 110, approximately then. There we can see that this has a specific geometry that allows many proteins. The oxygen acts as a dater or acceptor, perfect as an acceptor of hydrogen bridging. And the NH, of course, the opposite as a dater of hydrogen bridge. Now you will say, and the residue teacher, who does not play the residue in all this, still does not play a role for the structure of the proteins, the secondary and tertiary structure that we are going to be explaining. The most important, the main role, is played by the peptidic chain, what we are also going to call the peptidic backbone. Of course, there are two tertiary structures that interconnect very well then. The backbone, the way it adopts, depends on these oxygens, and these NHS, the residue still not. Okay, or in much less measure, we could say that the residues play a role in this. The second has to do with this. Look at the peptidic link. Note the following. A normal simple carbon-carbon link is 1.5 angstrom, but the peptidic link is shorter because exact is the double link, and that is to be cut the length. How much is an angstrom? Remember the value, more or less. These are atomic scales then. Okay, and what is the value of 1 angstrom? Exact in the distance, but the specific value, okay, it is 10 raised to minus 10 meters. Then it is a very small distance we are talking about to see if it would be 10 to minus 8 centimeters, the 100 millionth part of a centimeter, and that would be the distance of a link. A common link and an angstrom and a half, and this is shorter due to the resonance effect. Then we go to the structures that can adopt the proteins, and the structures we are going to classify them by level as they follow to see. We already explained that this drawing here more than the same thing that I just said already, to see short-term memory as this. Round symbol was called, and the round is written like this, and the other was the yes, I am like a pea forward. That is why the psychology school shows a lot of this symbol. Yes, of course they place. I've seen in school, in psychology books, I've seen that symbol. And there you caught me, that has to do with protein, excellent, well, in the equation of... Yes, exactly, yes, but hey, I'm not going to write it, but it has nothing to do with protein. Okay. Yes, of course it would be like this. This is the equation of Trenninger, the Hamiltonian operator, on the general wave function. The energy and the orbitals are ready to see. Then it turns out that in all protein the values of phi and of itself, in all protein the values of phi and of itself are fixed, because the proteins are not changing shape every time they maintain a way to preserve their function. If the shape of a protein changes, we would be talking of course, well, that concept of denaturalization, to see when we talk about the naturalization of a protein, it is for two reasons protein owe their shape to two variables, which are those variables that allow the shape of a protein, and if they change the shape, it is drastically altered by denaturalizing itself, by losing itself. Function, perfect, very well, temperature and pH were at a specific temperature and pH, very well. Now, having already explained the first 3 points that I have already developed, it is necessary to understand that the structure or folding or doubling of a protein depends on the formation of a hydrogen bridge between the NH of a peptidic link and the carbonyl of a peptidic link. This representation is not the best, because remember that the electronic pairs of oxygen are angular, they have angles of 120 degrees. So in reality, the proton reaches some of those angles of 120. Not in this way in 180 already, but the hydrogen bridge is understood, and for there to be a good hydrogen bridge apart from the exact angle of 2 to 2.5 anchors fluctuate the majority of the hydrogen bridges. Of course, because 3 anchors or more are already far away, and there I could do it clear of carbon alpha, maybe. We would have to measure, and that we can measure, and from there, we are going to do some, or I'm going to show you how we see this for free directly. And then we talk about first of primary structure when we refer only and exclusively to the sequence of a protein. And then, for example, I am going to ask you some questions here. Here there are several primary structures, several sequences to see. There is, for example, the following glutathione hormone. That stimulates the growth of tissues in the body, and well, that is generally stopped at a certain age when growth does not stop. We have problems right there were animals that did not stop growth. I do not know which animals were extinct, some of not all that never stopped growing. In fact, they died, and if until the last day, they were always ways growing, so to speak, the dinosaurs. If a certain type of dinosaur, as far as it is known as the Brontosaurus, for example, what can be known from a couple of bones, then that is that they grew all their lives and died like that. And that is why they reached those sizes so large. They grew in a proportional way. That is the interesting thing. Not all of us know that of the dinosaurs, of what size they were. I do not know if you know that the size is estimated between a chicken to a sheep. In that range of size they move the largest. Where the exceptions, And they were these, that as I tell you, that they grew and grew until we have several amino acid sequences here, the growth hormone, the hypothalamic hormone, TRH, the angiotensin hormone 2, which is an oppressive glass bradyquinine that exerts the opposite effect, is hypotensin hormone the oxytocin, which is the hormone that generates butyrate, has another function to regulate the mood, I think, thanks to. Not true, somatostatin inhibits it. Inhibits, it says, that the release of growth hormone endothelin, an oppressive hormone, methylene, glucagon, and insulin. Good question. Within all these sequences that are shown here to see, for example, we see the structure of insulin. And here, what we explained before, that they were two chains, but they come from only one gene. Here we have the sequence of all the amino acids that make up the insulin, the two chains that were interconnected and that were cut off. Post-translational question, there is the amino acidic commutativity to see. It will be the same to say to the ninoglycine, that connecting glycine to the nina will give her the same result when making the peptidic link does not give the same result. And there is no commutativity in the union. You alter one of them, and the function is lost, or the function is altered. Okay, and many genetic diseases are for that. Sometimes it does occur in some mutation. Then there is no commutativity in the union. Sometimes it does occur that some amino acids for example, suppose you that this nina is not so relevant for activity in the hormone. That was up here, which was this glutathione, not the glucagon. Okay, the hormone that makes the plasma release glucose. Then suppose that this nina is not very relevant. Good, if it and if it changes, for example, by another as leucine, which is also hydrophobic, maybe it doesn't function so much when there is a change by an amino acid of similar physical properties, but generally, the alteration of an amino acid implies that there is a damage to a coding for that amino acid, and that generates a disease, a defective protein. Well, then there is no commutativity in the union. Let's see now other questions related to the nomenclature of this question to see, for example, what it means in the case would be the methylene. Let's go here. It says bee venom. Then it means that this is the more protein that causes so much inflammation and pain. When someone has ever bitten a super painful and extremely painful, you know what it is for to immediately put your hand in something very hot. At least it works for me in a very hot compress. It helps me a lot to reduce the pain. And once it bit me here in the neck, and there it was desperate. Well, look here. We have this termination that you believe means because these chains have a convention to be written, because there is no commutativity. Then, Christopher, what goes to the left, the right? What is the convention? Remember here, it has the book, because if it is glycine, it has two arms. Okay, of course, here it would be from the synthetic point of view, or that we would have to have the free amino group to the left. Then it means that this glycine here has as free an NH3 plus, and therefore it means that at the other end a carboxylate was left over. Then ask why it is written like this. What does it mean here? A modification that means that the carboxylate was left over effectively. And look at NH2, said then, the carboxylate was left over, but post-translational modification that transformed the carboxylate into an amidite. That is why it said NH2. Okay then, if the carboxylate was there but within the enzymatic machinery of the bee is the poison of the bee. Right here, It was stuck, so to speak, an amine and iti became an amide, and then there was a post-translational modification that had to be explicit in the amino acid sequence, and down here, much more compact, the code of A. Single letter, well, the last one that means, for example, it is explicit here, this orange, yellow, a little yellow already, very well bridge and sulfur, then the oxidation occurred between this and this. And inside there are two more that with that question, how could it be formed if they are so far? As it is because the planes can rotate, then there is an explanation, very well, secondary structure by the primary structure, the amino acid sequence. Encoded from the genome, you know that this comes from the genome well. And we have that now the secondary structure comes from the formation of intramolecular hydrogen bridges. Intramolecular hydrogen bridges example. I am going to maximize this part of the protein. This is a type of O. Secondary structure called helix, specifically it is called helix 3 sub 10, and it is a folding in the form of, literally in the form of helix. That is because of the intramolecular hydrogen bridges that are formed between carbon dioxide of the backbone, NH of the backbone, not the residuals question. That means because there are two things implied here. Very well, okay already. Yes, that's right, 3 amino acids, very well. And the 10. As is 3 amino acids, and the 10 is the amount of atoms involved in a hydrogen bridge. I explain. I am going to count from here. It means that between this proton to the oxygen, between half here, there are 3 amino acids and 10 atoms then. If we count from the proton, it would be 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Then that comes from that 10 that we have here. There are 10 atoms to form the hydrogen bridges. And in between, there are also 3 amino acids, and this is repeated and repeated, and that is why the helix has a standard folding. Over time, well, with this, I think, we are going to finish for today. You have to explain then why this is fractional 4/416, that we quickly review the other class.